Hello. I'd like to tell you about my recent experience submitting a manuscript to F1000 Research. The manuscript in question was written by Amna Chowdhury and myself. Its title is Identification of Two Telomere Proximal Fission Yeast DNA Replication Origins Constrained by Nearby Cis-Acting Sequences to Replicate in Late S Phase. That's a complicated title, so let me explain. The issue we were trying to understand was control of the timing of telomere replication in fission yeast. In both fission and budding yeasts, telomeres replicate very late in S phase, and in both yeasts, this late replication requires an active replication checkpoint pathway. In budding yeast, late telomere replication also requires the physical structure of a telomere, that is, a chromosome end. In budding yeast, therefore, circular plasmids containing replication origins and telomere DNA sequences, but no ends, replicate early because they do not contain a physical end. Only linear plasmids, having two physical ends plus an origin and telomere sequences, replicate late in budding yeast. Whether a physical end is also required in fission yeast was not previously known. Our manuscript reported identification of the DNA replication origins located closest to telomeres in fission yeast. Our manuscript also described how circular plasmids containing these origins, plus about 6 kb of additional flanking telomeric DNA sequence, but not the physical ends themselves, replicated very late in S phase, just like the real telomeres on the ends of real chromosomes. But if the flanking sequences were removed, then these circular plasmids, now containing only the origins, replicated early, not late, in S phase. When the flanking sequences were present, late replication of the circular plasmids required an intact replication checkpoint pathway, just as in real chromosomes. Thus, the results of our manuscript, the results presented in our manuscript, demonstrated that the telomere proximal DNA sequences flanking the telomere proximal origins in fission yeast are responsible for checkpoint-dependent late telomere replication. This is different from budding yeast, where the flanking sequences themselves aren't sufficient to render telomeric origins late replicating. In budding yeast, physical ends are also required. Perhaps you can tell from my enthusiastic description of this research that I consider it to be quite important. So you might ask why I chose to publish it in F1000 Research, a journal that was just starting up at the time and therefore relatively unknown. There are several reasons. First, I'm in the process of retiring from research on DNA replication and moving to a new interest of mine, understanding and preventing global warming. So I wanted to select a journal that would publish our results quickly, thus permitting me to move on more rapidly to my new interests. As I'll describe in a minute, F1000 Research certainly fulfilled my expectation of rapid publication. I want to emphasize that by rapid publication, I do not mean simply putting our manuscript online, which F1000 Research does even before a manuscript has been reviewed. I don't consider a manuscript to be fully published until it has been thoroughly peer-reviewed. As I'll describe in a minute, F1000 Research accomplished the whole process, getting our manuscript online and getting it peer-reviewed much more rapidly than was the case for any of my more than 100 previous publications. Second, I wanted to publish in an open access journal. I feel frustrated by the pay-per-view model of many traditional journals. It's especially frustrating when I want to look at the online version of one of my own old papers, and I find that even I, as an author on the paper, need to pay a large fee just to see my own publication. Third, for a long time I've felt that the traditional peer review process with anonymous reviewing frequently permits abuses. Those abuses range from taking an overly long time to write a review, to writing a very hasty review, without having carefully read the manuscript, to intentionally finding trivial faults with a manuscript just to delay its publication. F1000 Research is the first reputable journal that I know of to wholeheartedly accept the concept of signed reviews, and I wanted to support this much-needed innovation. So, how did things go when I actually submitted my manuscript to F1000 Research? The online submission process was somewhat easier than the online submission used in most journals. Instead of needing to submit through a website with complicated forms to fill in, all I needed to do was email what I considered to be a final version of the manuscript in RTF or Word format to research@f1000.com. I then received prompt responses from the F1000 Research editorial staff, guiding me through the process of providing all the necessary information and data in their correct formats. These email interactions with the editorial staff were similar to the process of manuscript markup and editing that normally goes on after a manuscript is accepted for publication, after the refereeing process is completed at most other journals. In this case, however, due to the F1000 research policy of immediate publication, the markup process precedes peer review. I received final proofs from F1000 Research just 15 days after my initial submission. Due to illness on my part, I wasn't able to proofread those final proofs as rapidly as I would have liked. Despite that delay, my manuscript was published online and available for the entire world to read just 11 days after I had received the final proofs. Peer review started at that point. My 3 peer reviewers were world-class experts. Hisao Masai, a leader in the understanding of replication timing both in fission yeast and in human cells, also a leader in understanding other aspects of DNA replication. Toru Nakamura, a leader in the understanding of telomere maintenance and stress responses in fission yeast, and Jörg Behler, a leader in genome-wide functional analyses in fission yeast. The first review was received just 3 days after publication, the second after 10 days, and the third after 13 days. I'm pleased to report that all 3 reviews were favorable. 2 of the reviews included some suggestions intended to increase the clarity of the manuscript. I'm intending to submit a revised version. It will be version 2 and will become the default version, but version 1 will be retained on the website so that interested individuals will be able to view the full history of the manuscript. I want to point out that the total time from initial manuscript submission to final peer-reviewed online publication was just 1 month and 9 days. Usually one is lucky if the initial phase of peer reviewing is completed in such a short time. Publication itself usually takes a couple of months more, at least. In my experience, therefore, the F1000 research model provides the fastest submission to peer-reviewed publication time of any journal of which I'm aware. Overall, from start to finish, I have indeed been very happy that I decided to submit my manuscript to F1000 Research.